Scientists have turned individual cells into miniature lasers by injecting them with droplets of oil or fat mixed with fluorescent dye that can be activated by short pulses of light. This finding could help to broaden how light is used for both medical diagnosis and treatment. The system was devised by Harvard Medical School scientists, and it uses droplets of fat or oil within a cell to reflect and amplify light, generating a laser. Conventional luminescence probes, which include fluorescent dyes and proteins, have relatively broad emission spectra. This limits the number of probes that can be used simultaneously, because it is often difficult to distinguish these sources of luminescence from the broad background emissions of naturally occurring molecules in tissue. The microlasers could change this because their emission spectrum is narrow, making it easier to label cells with light. One of the greatest implications of the work is to track thousands of cells simultaneously with a single technique. The researchers report that they can vary the wavelength and tag individual cells using fluorescent polystyrene beads of different diameters rather than injected droplets of oil or fat. In theory, using different combinations of beads and dyes with the different spectral properties should make it possible to individually tag almost as many cells as exist in the human body. This technique is not yet ready for therapeutic use, but eventually the modified cells could be used to locate target tissue, such as a cancerous tumor, and active preloaded light sensitive drugs only in that area.